So maybe before we get started, just a few reminders of things that I mentioned earlier today. Just want to make sure that everybody hears. So we have Jupyter Hub. The hub is going to continue to be available to us for another 2 weeks from today. So if you're still in the middle of working something and you're kind of like want to keep working on the hub, you can do that for the next 2 weeks. But then it'll shut off, and there are instructions in Slack in a pinned message about how to get stuff that you have on the hub off of the hub. And if you try that and something goes wrong, then definitely send a message and try to sort that out. Speaking of which, the Slack will revert from its current paid version to the free version at the end of August. And that means that older messages will start disappearing off of Slack at the end of August. And that means that if you have like messages that are important, notes about things you're working on, things like that, put them somewhere else. But Slack will continue to be there, and we'd be happy to hear from you. You know, we can hear from each other, of course, through Slack. So stay in touch. That's all I'm remembering right now. And with that, I just wanted to say that it was a real pleasure to see you all working this week. It was really, really fun to see all the different projects and people getting off the ground with various things and then struggling with other things. And it was just really, really fun to see all working. And I'm really excited to see all the project presentations and to hear the stories that you were about to tell us. And with that very brief introduction, we will start with. Okay, yeah, the protocol is whoever's presenting will go on Zoom. If you're in the room and you have the laptop on which the presentation is, you will go on Zoom and share a screen. If you're already on Zoom, then you will share your screen on Zoom and show the presentation. And that way we will all see it on the screen here. Okay, and with that, we'll start with using machine learning to explore emotional states in the Human Connectome project.
Sure. Can you guys just confirm if you can hear me well?
Yeah, we can hear you well.
Right, okay. I'm going to share my screen. And again, just let me know when you can actually see my screen. All good. All right.
Hang on just a sec before you start.
Yes.
Okay, now you can go remove the obstacle. Go ahead.
All right. Thanks for the introduction, Ariel and Noah. We hope that all of you have had a very terrific hackathon. We really, really enjoyed our experience, and we are super excited to share with you some of the results that we produced. Our project tonight, or today, for those of you who are over there, is entitled Using Machine Learning to Explore Emotional States in the Human Connectome Project. So without further ado, I'm going to start with a brief introduction as to why psychopathological disorders matter, because we all know that they do. We know that globally, over 400 million people experience psychopathological disorders, and we know that these typically arise from enhanced negative emotional states or affect or loss of positive emotional experience. And we also know that younger adults are particularly vulnerable to these conditions. A lot of neuroimaging modalities such as functional magnetic resonance imaging and diffusion MRI have been used in addition to behavioral experiments and measures to leverage and assess disordered emotional states. And we also know that neuroimaging studies implicate a role for the limbic system in emotional processing. And here on the right-hand side, you can see a few of the brain regions that are crucially implicated in the processing of emotions in the brain. And that brings me to the project aims for our hackathon project this year. And that was to develop machine learning models to predict self-reported measures of emotional states from structural, functional, and diffusion MRI data using the Human Connectome project.
Okay, as mentioned, we did 3 modalities of the neuroimaging data: structural MRI, emotional processing task-based fMRI, and diffusion MRI. Specific regions of interest that we selected were chosen based on their established involvement in emotion and emotional processing by previous research. These ROIs include the amygdala, insula, anterior cingulate cortex, and medial orbitofrontal cortex. Given the bilateral nature of the emotional processing regions, average values were computed across hemispheres for each modality.
And then we used a variety of what we're calling here behavioral data, but is really self-report kind of survey data. And so I'd say primarily we focus on this first item here, the NIH Toolbox Emotional Battery, which is composed of a few different factors, including negative affect, psychological well-being, social relationships, and stress and self-efficacy. So each of the participants in this large data set completed items that spoke to each of these different factors. That's the one we'll really be focusing on, in addition to the 5-factor model of personality, which is, of course, this big, survey of personality that has these 5 factors. And then we put both of those together in a machine learning framework where the inputs involved primarily the neuroimaging data, in addition with some of the personality data. And then we used a variety of different models that we'll talk about. And the idea was to try to predict the responses to this emotion self-report NIH Toolbox battery. Next slide. So we can chat a bit about the results. So first of all, this is an analysis I undertook. And so we had, a lot of responses to different items for this NIH toolkit. And so my approach to simplify things a bit, right, this is supposed to be our outcome for the machine learning model. This is what we want to predict. And so my approach to doing this was to run a clustering analysis. And here I'm just sort of portraying a very simplified version of that. We have well-being on the y-axis and negative affect on the x-axis. And we were able to cluster subjects into 3 groups. That's what the analysis told us was the best fitting number of clusters. And it's nice that we kind of have a split between subjects who are high on well-being and low on negative affect, and then conversely some subjects who are high on negative affect, low on well-being, and some subjects who are neutral. And so this was ended up being the target for our machine learning prediction problem, was to try to predict for each subject which of these groups that they would fall into. Next slide. Okay, and I think Ray is gonna do this one.
Yeah, thank you, Dave. So apart from the 3 clusters assignment, we're also predicting the actual score from the 5 different emotional patterns, and also the PERT scores, which I think containing 5 different aspects. It has the output from 0 to 9, and here you can see some different models listed from the simple one to more complex one. And we have the logistic regression model developed by Dave, and it's taking all the data into consideration. Of course, that exclude the functional data because we haven't been able to hold that. And we also have MLP model doing the classification and regression. We also have like a multi-model GNN, which I'll show you the detail in the next few slides. Okay, this is the logistic regression part. I think I'll hand it to Dave.
Yep. So this is the analysis I did. So I ran a logistic regression, and here we're looking at 2 different metrics of performance, both the accuracy and the ROC score, which is a little bit more sensitive to the true and false positives of classification. And so I fit the model twice, once including only brain data, and once including all the data, including the responses to the Big 5 personality tests. And the distribution you see here, because we're social science researchers, we have to have a null distribution to understand anything, right? So to generate this, I just shuffled the labels and ran the classifier on nonsense labels 1,000 times to generate what we should see if guesses, our predictions were truly a chance. And so, maybe a little lesson about looking at different metrics of performance here. If we look only at the accuracy, we see that both the models modeling all the data and the brain data perform significantly better than what would be expected on chance. However, for the ROC score, we see that when you only include the brain data, we're actually right in the middle of that null distribution. Whereas when you include all the data, including the big five, we're doing quite better than chance. So yeah, it would have been nice to see like if we could predict these self-report responses from brain data alone. But it looks like based on this, predictions were pretty weak in that you include those personality characteristics, then the model performs much better. Next slide. Taylor.
Yeah, so for more complex models such as MLP or GN, they are hard to interpret. So you actually need to do XAI technique to analyze the importance of different training variables. But I'll just give you a brief introduction of how we're modeling the data from two modalities, the diffusion MRI and also the free surfer structural data. So for each node, we have different brain regions, and if there's like connection, we'll build an edge and give the weight for the strength. And for each node, we'll store the thickness or volume as the node embedding. So node embedding is actually some additional information to explain the information for each node. And after we build such graph for each sample in the dataset, we'll continue with the graph classification task, and then we're able to map those graph into the distinct labels. It could be PERT scores, or it could be the 3 clusters we have populated. Next slide, please. And yeah, overall, here's some major takeaways. So the challenges, first challenge is that our model has converged with pretty low accuracy or R² score. And second one is we're not able to download all the functional data and pre-process that. And so the key takeaway is why this how could we address the challenges is we could access more sample because we're kind of limited by the sample size. So in a machine learning standpoint, 112 samples might be still be small. And also we're not sure there's like a direct correlation between the training task versus the emotional status we're predicting. And but one thing is pretty valuable is we create a graph structure for both modality, which we could use for other predicting tasks. And yeah, next slide, please. Okay, I guess Dixon will continue from this.
Yeah, so now I'll wrap up everything. So what you guys see right now in front of you is a word cloud, or not really a cloud, a square cloud. But essentially, these are kind of like all the words and key phrases that all of us have kind of came together to realize and reflect on our experience with the Neurohack Academy program. And, next slide, please. I think what really fascinates me is first, like this is my first hackathon ever, so it was really cool to kind of see the fast pace of how things worked and how everyone collaborates and work together. But what really fascinated me more was that essentially the group that we're working with kind of spans across 4 different time zones, 3 countries and 9 US states. So the fact that we were able to kind of achieve kind of like a common goal within, the span of a week is just fascinating to me. So overall, I just wanted to say thank you. Thank you for listening to our presentation. And the QR code is a link to our GitHub repo in case anyone wants to check out any of our code and documentation. So thank you.
Great, thank you so much. The next group is eScience Busy. clear line down here and then join the Zoom.
Oh, okay, I need to do that. What do we do?
It's in Slack and
Tee, okay. Oh, gotta have one.
in general pretty low down somewhere.
No.
There. I'm trying the audio.
Is it going? No. Share. Engines.
All right.
Actually. Okay, you want to start again?
Yeah, this is great.
Oh, okay.
Where you can see how.
Okay, cool. Hi everyone, my name is Ashley and this is Naive Poppy Effects Viz. I did some work with Alex and Haley and also Hallie and Thomas online. So we have like 3 different projects in one. So that's what you're getting. 3 projects for the price of 1. Okay, so my end was to, edit brain effects a bit. So Hallie introduced it earlier this week, but it's a tool for exploring effect sizes in typical neuroimaging studies. And, I saw that and I thought it was cool, but I wanted to make it more user-friendly. So I wanted to add tutorial menus, I wanted to add tooltips and other bug fixes. And this actually gets us to our first demo. So let's do that. Okay, so this is what brain effects looked like before. Pretty typical, pretty standard stuff. Like you can affect, select your data set that you wanted, filter by map type. This is all of the stuff that was developed by her. But I saw this and I was like, okay, let's make it more user friendly. So that's where we get some pop-up tutorial menus to explain what the app is doing and stuff like that. So now it greets you with the welcome to Brain Effects and how to get started. And it like shows you to the menu to the left where you can select all the different things. And then you go next, and then it says, okay, you want to understand some plots. How do you do that? It explains how you would, and it points you to the plots in the middle and the right, and some additional information. And then you want to download your effect maps that you just filtered by. So then it explains how to do that there. So you close that window out and now there's tooltips that explain everything, data set to visualize, stuff like that. And if you ever forgot how to use the app, you can click on the button again. The other thing I did was I made this dynamic menu, which changes as you change what you're filtering by. And that's kind of useful because, if you did a lot of selections and you don't exactly remember what you actually plotted, it'll just remind you like you're looking at all data sets, the functional connectivity map type, and then if you load a task, it'll load a task for you and display it there. It takes a bit to load. But yeah, oh, and I also added the lab logo. And I think that's important. So yeah, that was my end of things, but I'll let you guys take the wheel.
Okay, yeah, so Hallie and I worked on the next part, which was trying to make workflows a little bit more reproducible. So if you're anything like us, you might have a script that has multiple functions that are in the script. You might lose some track of which function comes first and what's the input and the output. So our idea was to make a function that could track that for you automatically. So we created script to viz using Mermaid.js, and how that works, it's JavaScript, and nodes are the squares or circles, and they connect via edges, and those are the lines. So it shows the direction. You'll have a script and the direction of the input and the output. And why would you want to use this? It helps, automated generation, standardized workflow, and also version controls. There's a bunch of really cool features about this. Yeah, so I'm going to give Hallie the mic, and she's going to do a demo.
All right.
Hopefully this works better than the last time I tried to do a demo. So here we are. Okay, so this is the repo for the project. And if you wanted to use it, what you can do is actually, I'm getting ahead of myself. Okay, this is what it does, is the end product is that in your GitHub repo or directory that you have, if you have a README file, you can automatically generate this workflow diagram of a script in your repo. So you tell the function what script is the one that you want to visualize. It has all your functions in it, and then it makes this from that. So you have the inputs in red here. Look red, yeah. In red, these are the functions in green, intermediates, and then your outputs at the bottom. Then with the GitHub workflows, we made it so that if you make a change to anything in the repository, it automatically goes and updates this and puts it back in here in an updated version. So if you wanted to do this, you just download this script and the workflow, and then you just have to edit the script to say what the name of the actual script you want to visualize is. And yeah, I think that's all for me. I'll put it back to the slideshow and we'll move on to MLS's version or project. Anything
Thank you. Thank you. Yeah, so my end of the deal was to try to get more data into brain effects, and we were thinking of, yeah, a pipeline, a BIDS app, basically, that could maybe take a BIDS directory that has, yeah, for starters, fMRI prep derivatives, and depending on whether it finds that there's task fMRI data or resting state, it could calculate connectivity maps according to a pre-specified template, and yeah, do some group level calculations, and maybe one day send it to a central repository. Could I ask for next, please? Yeah, so I only had time at the moment to work on the resting state stuff, so the task from my part is still to be done. And next, please. The point would be, or the big vision here would be that this could be a BIDS app that could be run on basically all open neuro data sets and basically calculate effect sizes, simple effect sizes for all data sets that have necessary files in them. Thank you. Next. Relatedly, we also started thinking about an exciting topic of how to design kind of like BIDSified second order, third order derivatives. And actually, this is still seems to be an active point of discussion within the BIDS community. And there's a couple of BIDS extension discussions going on that people can still join if they have thoughts on the matter. Awesome. Next, please. Cool things I've learned was, submitting issues on my own repositories, asking people who are more knowledgeable than me to help. So for example, here's an example of Michelle helping me with a bug that I was having in iPuppy, and that was very cool. And my last slide is that, yeah, we wrote some cool functions here that seems to do some basic resting state connectivity, and we got out some first order and second order derivatives. So it's exciting times. Thank you.
And thanks, Steph, JB, and Michelle for all of your help. That's all.
Thanks so much. The next group up is the NeuroNest group. Who has the slides?
There are no slides.
No slides.
No slides, it's the website.
Okay, should we show it from this computer or, yeah? Okay, then we have to share the screen, yeah?
Yes.
Yeah. Okay.
Just you just want to go?
Yeah. I know it's a Neurohack GitHub.
It's
I have access to the GitHub.
Whoops.
Like the Neurohack, I just
yeah,
don't
yeah.
remember the.
You don't remember your repository name?
Their academy.
There it is. We
Whoops,
found it.
Neurohack.
Yay, I know I put it
It's
somewhere.
like
What's the name?
neuroacademy24.io, and then it's gonna go.
Oh,
Yeah.
got it.
Linky foo. Neuronest/index.
Okay.
Yeah.
That did not come up with anything.
Right.
Backslash neuronest,
okay, okay.
backslash.
Yeah, yeah.
We're so close. Neuronest/index.html. Yeah.
There you
Okay, you need to probably share.
go. Oh, you're, yeah, sorry.
I'm so sorry.
No problem. This is a Zoom problem, not a you problem. Where is that?
That's
No, yeah, that box.
All right. Hello, everybody. We work on something called Neuronest. So I think everyone may or may not remember from the very first day whenever we did introductions, I had a thing about geese. So I tried to go with bird theme. So what is Neuronest? Neuronest is a website that allows you to access lots of information about lots of different topics that are all imaging and data science kind of focused. So Ariana is going to be our Vanna White, and she's going to click around while I talk a little bit. So a little bit about me. We're going to start there because all great stories start with somebody. It's me. When I got my job where I currently work, the very first thing I did is I said, I have a background in data science. How do I make this work with neuroimaging? And the only resource that anyone pointed me to was actually Ariel's book. And while that was great, it didn't fulfill all of my needs or answer all of my questions. So I'm here at Neuro Academy trying to get my questions answered and my needs fulfilled. And I still feel like maybe there's a gap, and maybe you guys feel that way too. So we created a repository of information. On the front page, there are many tiles. The ones closer to the top are going to be imaging modalities. I personally have no experience with about half of these. I'm sure you might not have experience with them either. So if you're interested in finding more information, you can click on one of the tiles, you can go into the page, and you can read a little bit about generally what it is, what does the data look like, and why would you work with this type of data? What does it work well with? Is it MRI? Does it work well with CT? Does it work well with EEG? And then let's say you're like, I know what imaging modality I want to work with, but I'm not sure what kind of analysis I want to do. You can go back down, you can scooch down, you can say, do I want to work with MATLAB? Do I want to learn more about Python? Maybe I want to learn more about data pre-processing. Or if you're feeling extra feisty, we've got some command lines, some GitHub and Docker, some open scientific data sharing. Down at the bottom, we have advanced analysis, so it's going to be like MRI, meta-analysis. We also have alternative programming languages. Personally, I've really been getting into Julia, and maybe you're interested in learning more about Julia. And we have contribute. That's instructions on if you wanted to update the page. We also have some additional, you can click on the hamburger menu, please. We have ask a question and an about page. So Ariel's gonna go to the ask a question page.
Yes, right.
Thank you. Here, you can post your own information. Let's say you've been doing an analysis. You're like, hey, you know what would be great? Somebody else knew what I was doing, so they didn't have the same problems. Bam, a place to do it. So we worked really hard. The whole team actually took about 2 tiles each. And they worked really, really hard to kind of put this together. And I'm really grateful for the team. And that's why my team is now going to come up and including the online people and give me quick, what did they learn?
Yeah, I'm truly excited to have been part of this project because our aim was to combine the knowledge acquired during these weeks and transform it into a resource useful for everyone. So when I was young, I always dreamed to open my own museum and yeah, make the knowledge share with others. So why this project isn't exactly like opening a museum, I think that the underlying interest is the same. So science shouldn't be for a few, but should be shared with others, making more complex ideas more accessible for all. So I think that this project is in the right direction.
Yeah.
All right, I'll keep it short. So yeah, I wanted to work on this project because I wanted to help create a site where there was accessible data, and like tutorials and information. Because when I was introduced to a lot of these topics, like somebody taught it to me, but it still didn't kind of click. And it would have been really nice to have kind of a central area where you have different resources linked to each page. Also, working on this project gave me the opportunity to kind of review and learn some things that I hadn't. So I had put together like some Python page, the MATLAB tutorial gave me practice with SPM, which I haven't really touched in a hot second. Yeah.
Hello, is this close enough? I don't know. Yeah, this was a really great project. I'm really excited that we got to work on it. Obviously, it's like an incomplete project because it was only a few days, but I think that's part of the exciting part is we get to keep working on it. And I was just thinking if there were any resources that y'all had that were particularly useful for you when you were learning, or if there's anything you would like to contribute, please let us know. We'll really let Cindy know, and she can hook us up with anything that you have for us. But yeah, it was a great project and hopefully it'll be useful in the future.
And then we have some friends online if they would like to share.
Well, my name is Alan, and well, I wanted to join this project, this tutorial, because well, I'm a psychiatrist and did my master's degree in fMRI analysis, and when I began studying, it was a very different topics from my background, so I had a lot of difficulties, struggle with pre-processing and different tools about MRI analysis. So this idea looks wonderful for me because it's like give like a resume, a combination of all the different tools, tutorials, and tips about coding that I found it was difficult for me to find back then. So thank you a lot.
I can go next. Yeah, this was an incredible feat of a group project because this is a big hybrid group and it was very productive and very cool to see everybody bring their different knowledge and kind of come together. And I learned a lot from other people. I got to, refresh my mind about how you do EEG pre-processing and wrote a tutorial for that. And that was a great reminder of all the Python libraries that people typically use and the methods, and to be able to explain it to somebody who would be a newbie was really fun. And to also learn, from the tutorials that other people wrote was very informative. So it was a really fun learning experience and also knowledge refresher.
I think it's all
Here, I'll go. Hi, everybody. So I came in as a newbie and I went into areas that I was not familiar with at all, which one was the scalp EEG. And also very synchronously, I actually was able to shadow scalp EEG this week, not planned. So for myself, what I realized, one were some gaps. One is I really wanted practice questions, or I wasn't finding that, so I created my own. So that's for this, which I thought would be helpful. And yeah, overall, I would say there's definitely a need in the scalp EEG space to have something short and concise that has like practice questions. So that was helpful to have that insight. And then also first time with Python ever. So matplotlib, I was able to have practice with that. It's not linked in yet. Still figuring out how to do that on the site, but yeah, so great learning.
I think that's all of our online friends, right? Wonderful. All right, that's everything. Thank you.
Thanks a lot. Next up, I'll stop sharing here. And I will hand it off to the use case of QSI prep team. Yeah. Oh, sorry.
Yeah. This- Okay.
Yeah. Wait. Yeah, I think it will be easier like this. I think we won't have to, but yeah, thank you very much. So hi, everybody. We are the diffusion team, and we did a use case of QSI prep. So our main aims for this week were to test using QSI prep for pre-processing and reconstructing diffusion MRI data. And in parallel, Luis and Gabby teamed up to compare different reconstruction models by comparing a simply DiPi pre-processing pipeline with QSI prep and comparing diffusion tensor imaging with constrained spherical deconvolution models. Yeah. There you go.
So we began our journey here, learning together about different topics in diffusion, models, and introduction to QSI, preprocessing and reconstruction. And we create a document that you could find in our report. So you go there and enjoy.
And this is just to orient you at sort of a high level as to what is exactly on our GitHub page. So we didn't exactly propose anything novel, but I think what we wanted to do, as we are all true beginners with QSI prep, anything diffusion, we wanted to provide sort of a step-by-step process of like what that would look like, and perhaps by providing issues we ran into, it would be helpful for other people in the future. And this is, again, just a high-level overview of exactly what the page looks like. It has sort of the background information on DSI, as Lupita just mentioned, as well as all the code and the scripts that we created, as well as sort of information about the data set that we used.
So next, after all being in line with the literature, we decided which data set to use. So we wanted to use an open source data set. We started considering HCP, but we figured that it is already minimally pre-processed. And it was not fully compliant to the bits format that QSI prep requires. That's why we moved to using the Healthy Brain Network. And if you ever want to use it, you can find under this link the unprocessed data for each subject and according to their site, where the anatomical, DWI, and the field map files are stored. And we created a script based on the data showcase lecture to download the data to the cloud. We worked only with 2 subjects, so we created a function to crawl through the folders of each subject to download them to our temporary folder. Yeah, and next.
So the first thing we learned when setting up our environment to be compatible with QSI prep was that we couldn't use Docker and Jupyter Hub. So the first thing we did was figure out the latest version of the Singularity image to create, and then we built that in our environment. And then the next thing we realized was that our bids wasn't exactly well suited for QSI prep. So first what we needed to do was basically remove some fMRI field maps. And then we needed to make sure we had the relevant bid structure, including the dataset description JSON file.
Yeah, just to orient those of you who might not know a little bit to the QSI prep workflow, this pipeline handles both pre-processing and reconstruction. It's based closely on fMRI prep, so it uses the best tools from many different software packages. Pre-processing only is the default, so unless you specify recon, when you're running it, you'll get pre-processing outputs. And BIDS is the foundation, so as long as your data files and the structure is truly in BIDS, this will run for you and it will customize or build a workflow based on your data. So we put together a script to run this in our Singularity. In the script, you can see we point both to the Singularity image, we point to our input data, where we want the outputs to be. We also point to a free server license because QSI prep pulls from free server to run some things. We also tell it that we want to run just one subject at a time, and then we give the resolution of our raw data. And here we just ran with the defaults, but you can certainly specify options based on your specific case. This would be most likely related to things like grouping scans, denoising, correcting for head motion, and enabling or disabling steps based on your needs.
After running the pre-processing, around 6 hours later, we end up with the well-organized folder structure of the outputs. The main folder contains an HTML for each subject, which is a visual quality report that tracks the changes at each step. And inside each subject folder, you'll find a pre-processed anatomical and diffusion image for future analysis. And next, we document the guide of the HTML output figure by figure, which I believe we are the first ones to do it. So check it up in our repository if you ever want to use that. It is separated in anatomical, denoising, and diffusion processing parts. Here I show 2 examples. After the bias correction, you could see that the image is more normalized and inner part has better intensity. And distortion correction also did a really great job in the prefrontal area. Yeah.
And once we were done with the pre-processing step, we proceeded with the reconstruction pipeline. And here, as Alessandra said, we also only use the defaults of QSI Recon, but the toolbox allows you to adapt it to your needs, selecting according to your acquisition schemes or whether you want to have a deterministic or probabilistic tractography or use different atlases. And yeah, we created a 3rd script to run at the subject level. Unfortunately, yesterday we bumped into several errors and we were locked out several times of the server, so we cannot present the results, unfortunately, but stay tuned. Just to guide you through it, it's based on the pre-processing script that Alessandra presented, but you additionally define the QSI prep path where the outputs of the pre-processing are, and the model that you want to use. And under the website, there are already a bunch of models created, but you can also create your custom models according to your needs. Yeah.
So even though we couldn't run QSI Recon, we still like try to do some like reconstruction. So to do so, we wanted to compare the QSI, how reconstruction will be comparing the QSI output versus a simple pre-processing that we would do based on like some packages. So what we did was pretty much we took the raw data, then we denoised the data, we did some motion correction, registered to the T1, and then we ran the reconstruction. So what I'm showing next is how the output looks like. So we have like the simple pipeline up and then the QSI prep output down there. I'm pretty sure we could like improve it, but this is what we got out of the simple pipeline. And then what we wanted to do next, we just wanted to test the models, the estimation models. So we took 2 voxels, one of them with parallel fibers and the other one with a lot of crossing fibers. And what we did next was we compared both the QSI output versus our simple like pipeline. And then we can see that for those with parallel fibers, for the voxel with parallel fibers, QSI prep was like really good at explaining the variance, but like for the other voxel with crossing fibers, not so much. So then what we did next was we tried to fit, instead of simple diffusion tensor imaging, we tried to fit the constraint spherical deconvolution. And then what we're looking at is just the QSI prep outputs, and we just like try to fit the different models, and we can go into the details of this if we have time later, but I don't think we have time. So
yeah, so wrapping up and going back to our aims, we were able to run the pre-processing pipeline, but the reconstruction pipeline is on the way. And Gabby and Luis were able to compare the different models. And yeah, along this journey, one thing that we could confirm is the programmer's credo, that we do these things not because they're easy, but because we thought they were easy, or they were going to be easy. And it was the small details where we were hanged up for a long time to get things running. So some of these small details, but which were big takeaways of this week, were first that we could not collaborate or work on our scripts in parallel within the Git repo, and that QSI prep really requires a bits compliant data set, and that includes the data description JSON file. We also want to highlight the relevance of the documentation because we identified a bug in the QSI prep documentation according to the flags, the argument flags to run the functions. And one problem where we were hanged up for a long time yesterday was in Bash that we were not aware that you couldn't include blank spaces or comments after the backslash in a continuous command. And also, Gabby and Luis were having issues running DIPy visualization functions in the Jupyter lab. And yeah, last but not least, we want to thank Ariel, Noah, and Kelly for having us here in the Neurohack Academy and to nurture this open and collaborative environment, because we truly believe that this really helps in the learning process and to create a very nice community feeling. And also thank you to our members online. Yeah.
Fantastic. Next, we will have the sleuth slot, TLDR, Scientific Journals and Articles team. Fair, that thing about the white space after slashes in Bash threw us off for a long while.
Share screen.
Fantastic.
All right, hello everybody. So we're introducing our app in progress for sure is the sleuth slot. So those are the too long didn't read for scientific journals and articles. Essentially, it's a search engine to search through, and if you ask me if prompted with a question, it will give you the specific articles you're looking for. So just to begin with, how we move this way. Okay. How we decided to break apart this project. So our main goal, after some discussion, was instead of doing a whole kind of web-based search of articles, like having a search bar to look through all these articles online, we decided to make it a local environment. So essentially, if you have a Zotero library or some kind of file explorer library, you'll have a search bar where you can ask it some kind of question about what article you're looking for. Oops.
I moved so it wouldn't do that.
One moment.
This computer somehow, but see if that cover right there has been.
It's coming back. It's all right. Maybe.
Yeah, it's coming back. It just.
So essentially, it will populate all the articles that you're looking for. And then we also wanted to include a visualization piece. So if you're interested in more information on a specific article, it'll kind of pop up a window and then give you an idea of descriptive statistics and things like that. So those are just our key components. That's a local search engine. We're organizing it in a table format at first when you are searching for this article, and then you can click on those individual ones to kind of pop up more information or a general summary. And then a big part of our first day and second day was splitting up the tasks, because we're just building from the ground up. This is totally from scratch. And that was a little bit ambitious on our part, and you will see that through the rest of this presentation. So we'll start out with Christian, who will just talk briefly about the user interface that he created.
Alrighty, hi everyone, I'm Christian. So for the user interface, I started out by deciding that we should do a standalone kind of desktop application. And so in order to do that, I started out using what's called tkinter, which is a Python package, which is, as I have been made aware of later, archaic. And so I decided to go ahead and use WX Python. I'm not as familiar with it, but it's a more modern package for using GUI designs. And so I consulted with a friend of mine called ChatGPT to help me translate from tkinter to WX a little bit there. And as for displaying the data frame that you'll see here in a moment in the GUI, I use pandas, the standard. And so that's just a little screenshot of one bit of code that there is, but this is kind of like the main GUI filter that you're greeted with when you first open the program. And then there's different, there's a file and import, you could import things, and there's a search bar, and well, there you go. When you import it, you get this is a sample data set that I made in 2 minutes to actually reflect what you would actually see. This is really demonstrative purposes. but yeah, so the idea is that it's an intuitive design, also work in progress, so that you can utilize things as you wish. So.
So yeah, as Loretta mentioned, we were first going to start with like, crawling through the entire World Wide Web, but then lowered our ambitions. And so first we, let's skip the slide. We decided to, just collect all of our data from the local Zatora library. So like hypothetically, a user would connect their Zatora to this program, and it would crawl through their database and it would extract all the metadata necessary for it to index these papers. So things like the storage key, the PDF file or PDF name, and the PDF path. And so that would be saved in a CSV file. We use these identifiers, which are the storage keys, to index these papers, because there's a lot of papers that you might have that are all from the same author, have very similar names. And then you'd convert those papers into text files, so that we could, facilitate this batch process later on. Ooh, sorry. Okay, can you hear me now?
Yeah.
Okay, so then you convert these PDFs into text files, which would facilitate batch processing. Of the 870 papers that I had, it took it like 25 seconds, so it was actually very fast. And then, yeah, I'll pass it on to you for language processing.
Okay, so I was focused on the natural language processing element, which I've never done before. So I was really trying to understand how computers are understanding text data. And I'm not going to go into too much detail because we have a lot of other slides. But these are some packages that might be helpful. And there were two primary ways of going about text analysis that I could see. And so the first one was using those top two images, so tokenizers, which is really just breaking down longer phrases and sentences into their individual words or like smaller subparts. And then word vectors, so this is just how computers take the semantic data and they're just like finding a numerical way to categorize how similar words are. And then the other method is embedded transformers, so this is a little more complicated. But essentially what you need to know is that it's just better at more complex semantic processing. So some key scripts that I needed to include, so tokenizing, which is the concept that I talked about earlier. So all of the words need to be broken up. And then the second thing is called named entity recognition. And so this helps the model recognize certain categories for words. So on the top right, you can see how it's organizing things by person or location. And so you can either use a pre-built version of these labels or you can customize one. And the last one was part of speech tagging, which that's kind of self-explanatory. It just breaks down the sentence into the part of speech of each word. So first I used Spacey, which is a really cool place to start when it comes to NLP. And I was using pre-built NER classification, but I ran into some issues when I tested this out with sample sentences. So I have the exact same code that's processing two different sentences. Sally speaks English and Kirti speaks a language called Tamil. So essentially they're saying the same thing. If anything, the sentence with my name is a little more clear because that's why Tamil is a language. But when it interprets it, in the first one, it clearly says Sally is a person and English is a language. On the second one, it says Kirti, I'm a person, but it says I'm an organization. And Tamil is a political group and a language. So anyway, I ran into some issues with that. So I had this idea of using BERT, which is one of the embedded transformers. And so I was trying to create like custom labels. So you can see in the left part here, I have a bunch of custom labels that also include things like neuroimaging would be separately pulled out. And so a lot of things that would come up in scientific articles were separate labels. And then, yeah, so then I had BERT align, so tokenize a sentence and then align each of the labels of the sentence. And you're supposed to feed it a lot of data. I was doing this manually, so I did not feed it a lot of data, and that's kind of where I ran into trouble. So the data prep part was pretty straightforward. Like all those steps that I talked about, I was able to get those accomplished. When I created the model and trained it and then tried to test it out with keywords, I was not able to extract one, like extract relevant sentences.
But Iden was working on something similar and had a little more success when it came to summarizing the papers.
So whereas Kitty had the idea of using BERT, I used BART. They're different. But so yeah, so we first started off with the document classification. So train this natural language processing model on sequence classification to categorize text sequences. So things like sentences, paragraphs into these predefined labels. So went through the first 40 papers that I had in my tutorial, just labeled them whether they used a behavioral method, neuroimaging, whether behavioral and neuroimaging. There was some computational in there as well as reviews. So I trained this model and spent a couple hours training it. And after training it on labeled papers, the model was supposed to classify these new texts and update the CSV with the predicted label. So you put in these papers, maybe they're labeled already, like to which category they belong, maybe not, and then it populates that method that they used. But you need to have a well-balanced data set, because if you don't, then it's gonna just classify all 800 of your papers as the wrong thing. And also 40 papers isn't enough to train for 800. So after it kind of scrapped that, so I ended up just continuing to use the first 40 papers for the rest of this. So the next process was trying to summarize the texts, and the way that worked, it was it would extract the abstracts and then clean the texts and lematize them. So it would first get rid of all the exclamation points, all the punctuations, numbers, really long spaces. And then it would reduce the words down to their like just base value. So something like running would be reduced to run, something like driving is drive. And then it'd use BART to generate these really concise summaries that are very beautifully written and super coherent, as you can read here. But it should work if you train it well. So these summaries would then be extracted and put into the CSV file. And then the next process would be the question, like the query. You pose it a question, it's supposed to spit out which papers it thinks you're asking about. And the way that worked is it would first convert those summaries into these numerical values using TF-IDF, which essentially measures the importance of a word in a document, and then compares it to the frequency across all of the documents, and use that tokenization method that Kirti was mentioning. And it searches through all the summaries and finds the ones that are most similar to your query. So here's a sort of example. Just type in correlative event boundaries, and it pops up with all the papers that are most relevant, or that it thinks are most relevant. So essentially, design a really bad search engine for your papers. Cool. I'll pass on to.
All right, and then so if we did have that figured out, the next step would be to have your data frame with all your tables, and the next step is to kind of try to visualize how this GUI format would look like. So this is kind of our brainstorming phase where I just made brief sketches. Magdalena and I worked on it, and we kind of came with this idea that once this data frame is populated, you'd be able to click, and then it pops up this window, and then it would have a selection of different variables that we decide to pick out from this data frame. So we decided to do like a title, author year, just a summary of essentially what the paper would be, and then have some descriptive statistics. And the plan was to use a GUI interface. So first I was trying to use WX Python GUI programming and miserably failed at that. So then I tried to do custom tkinter as well, and I was not successful there either. So I have no experience with the GUI interfaces, and that goes to show because I don't have an interface to show you guys. But ideally, this is what you get popped out. So I'll hand it over to Magdalena to talk a little bit more about that.
Yeah, so together with Lera, we spent some time trying to think of a nice and way to summarize visually these variables. And I was playing with Python a little bit. I'm new with Python, so I'm happy with the result. And we decided to use these two graphs that you see, like a stacked bar that represents the groups of the article separated by sex. And you can also see the sample size, and then on the right, visualization of age mean and range. And yeah, and I also learned a little bit about natural language processing. I'm super new to this field, so it was nice to just learn how
It's easier to extract metadata from articles with Crossref, for instance, but if we want to extract a specific demographic or methods, variables, or results, it gets more complicated, and we need to use regular expression patterns or some custom code, dependency parsing too. So yeah, it was nice to learn that. And some next steps, we thought we could do this pop-up panel a little bit more interactive, or it would be nice to maybe display some statistical results, maybe using the StatCheck Python package. Next slide. And yeah, after the pitch, we realized that there's this really nice tool that basically does the same that we are proposing. It's really nice. It doesn't have the visualization panel though, but you can ask a question and it will give you the most relevant papers, and you can like add columns with AI prompts with like specific questions like age, population, sample, and it's really nice because you can also modify the prompts. So yeah, I invite you all to check this tool for your literature reviews. Next.
Yeah, and with that, we did, I guess, to summarize, we learned a lot about a bunch of different types of resources that you can use. So if you guys want, we might post that out just to see all the things that we've actually found out with this. And then I think we did a lot of learning. So even though we didn't get together, there's a huge appreciation for search engines because it is incredibly hard to create. And I think we realized that after the fact.
There's so many words.
So many words. So many problems. Same thing with a GUI interface. Oh my gosh, I had no idea that it was gonna be that difficult. I thought I was like, yeah, I'll just make some buttons. It'll be so easy. No, it's not. So huge appreciation for what's out there. Use these resources, and thank you so much. Thanks for the class.
Thank you so much. The next team is No Scientist is an Island.
I think Megan's gonna share her screen.
Okay, good.
And I was wondering if it'd be possible to pin her since it's just her,
yeah.
she'll be talking a good chunk of the time. But
Yeah, let me do that.
as well. Yeah. Thank you so much.
Okay, can everyone see my screen?
Not really.
No.
Okay. I think this should work. We see you.
Okay.
We see your screen.
Okay,
Great.
great.
All right, you're ready.
Okay, so so Teresa and I identified a problem that we think many labs face, and that they don't systematically track the creation and existence of data derivatives that their members create or attempt to create while they're in the lab. And to clarify what we mean by data derivatives, because in other contexts this means something else, for our presentations today, we mean taking raw or minimally processed data and doing something to it to create a new variable. And so that includes things like what appear simple, like summing a couple different questions in a survey instrument, or creating a more complicated composite variable. But we think even these most simple summing of a few questions requires some judgment calls, like how you handled missing data, if you did any imputation. And a lot of critical information about the development of these variables is missing. It's either buried deep in code. You don't even know where it is if you went back to look for it. Even if you found it, you might not know exactly what you did or why because you didn't document it. And if someone else wants to use it, so say a collaborator or a collaborator of your PI emails you and asks you for this variable you worked on a few years ago, it requires a lot of effort to go find it, clean it up, make sure it's ready to share, and actually send it and hope that it was accurate. And additionally, someone else in your group, either simultaneously or a few years ago, might have already done this. So we're proposing a flexible structure that can be used by labs, small groups, or research groups really of any kind, and we're calling it a derived data registry. This is really geared for phenotypic variables, so tabular data, but it could be extended to others. And the target audience for this framework are groups that are working in environments with little or maybe mixed adoption of reproducible programming practices and different open science practices. So this isn't for experts, this isn't for people who are already doing this, this is for people who maybe don't really know what this means. So we came up with No Scientist as an Island to highlight how some changes to our behaviors around tracking and sharing data derivatives can integrate researchers who are working what might feel very independently, but are actually doing things that relate to each other. And so we think this framework can really help increase teamwork and prevent knowledge loss. There's a couple different reasons why we think this is important. So when people leave, you're retaining some of this knowledge they've generated, you're increasing reproducibility of your own science, and you're fostering collaboration.
So we created sort of a quick start guide to this, kicking off with just defining a derived variable. And in particular, we had some conversation about what kinds of variables are worth logging. We decided that a good general rule of thumb might be a variable that you are going to put into an analysis or model sort of building stage might be something that's worth letting other people know that you've taken the effort to create. We also came up with the idea of a derived data registry. It's just 3 components. It's pretty straightforward. It's a log, some code, and the actual derived variables that somebody else can access. So this is the kind of more detailed information about the actual setup. We sort of don't require researchers to set up their project directories in any particular way, but we do provide some recommendations for where the derived data registry should be. And then also we stumbled across an emerging specification called PsycDS, which was created in a hackathon by SIPS, the Society for the Improvement of Psychological Science, in 2018. And it's modeled after BIDS, but it's for the use of this more like tabular sort of data format. And we have kind of conceptualized our work as a potential extension of PsycDS, and maybe we might be in touch with them about contributing, because we think that for derived variables, they have a nice kind of specification for what the kinds of metadata you should track for variables in general, like what's the name, what's the description, what type is it, what's the max value, is missing NA or is missing 999, stuff like that. But we think that for a derived variable, there's actually additional information that can be really helpful. So for example, there's an author and reviewers. So that helps you keep track of who to actually talk to about these variables. A git show, so you can kind of connect to the exact commit where this variable was added to the registry. And then something that we call log status that we'll talk a little bit more about. I won't go through all of them, but this is the sort of idea of different things that you could, different types of metadata that might be important to track for derived variables.
Yeah, so we have this idea, how do you implement it? We came up with this big flow chart here of how you would actually use a derived data registry, and it's broken up into a few key stages. The first stage is really the conceptualization stage, then development, the actual creation of this variable, reviewing it, and then finally registration of the variable in the registry. And so on our site, we have pages for each of these stages. So first, this really gets at, checking the registry. I have this idea. Has someone else already tried to do this? Could I help them get it to the finish line? Did someone try this and abandon it because it wasn't viable, or there was something wrong? Using this log status in the registry to gauge whether or not what's already out there is useful for your case or not is really helpful. And so I've highlighted a couple of the different statuses that could appear under that log status field in the log file. Then the next step, once you've decided, okay, I do need to create this thing, we have a page about developing a derivative. And we've really geared this towards people who maybe don't have a very strong coding background. Maybe they're the people, when they make a derivative, are doing it in Excel, by hand. And we want to push them towards thinking about, yes, there is an upfront cost to learning how to script this and make it more of an automatic process, but it's really worth it in the end. This is one of our 2 key principles here, and that there's many long-term incentives for figuring this out. Transferable skills, you're going to reuse it again. Someone else could use it again. You're going to learn something new. But also we want to highlight, you should never modify raw data files and do stuff in Excel with it. So there's a lot of resources here. I'm not going to talk through all of them. A lot of this is stuff inspired by what we learned last week. And so really thinking about what level of detail you need to provide in your code, the comments, variable naming, code, formatting specifications. And we acknowledge that not all groups are going to just suddenly start using PEP8, but maybe being more consistent internally will make their codes more readable to their lab members in the future. We also provide a checklist at the bottom of this page to make sure your derivative is meeting some of these, suggestions we've provided before you actually move on to a code review. And I know for me personally, I don't think anyone's ever reviewed my code. So we read a lot about how do you do this? What's the point of a code review? Some best practices. We talked to Ariel about this, but we really want you to be able to explain your code to other people and just to get another set of eyeballs, ideally, on it. It'll probably make you a better programmer. It'll make you a better teacher. Many benefits of going through code review. And we've also provided another checklist for the code reviewer here. What should you be looking for? And then finally, once you've gotten through that, you submit it to the registry. And so it's not only to change the log status to registered, but also upload your code and that derived data to the registry itself. And we want to emphasize that the code needs to be modular. So it needs to go from some raw or minimally processed data, version stamp data to the derivative. And we want code per derivative in that registry, or code per set of derivatives if they're really related. And now Teresa's going to talk about implementation.
Getting a group of people to adopt a shared framework is super hard. And so we started thinking about, in practice, what would this look like? What might some checks be that could be tied to things that you're already doing in your research process? So for example, we have our workflow that we're proposing, but one checkpoint might be, hey, if you're sharing an analysis plan with your PI, with your collaborators for feedback, or maybe a pre-registration, when you get to that stage, that's a good checkpoint for thinking, hey, have you checked if these variables exist? If you're definitely planning on making them, maybe this is a good time to log them as under development, so you can let other people know that this is happening. Another checkpoint that I think is really wise is if you are kind of at the point of, submitting a manuscript or putting up a preprint, and the basic variables that you are putting into your models haven't actually been verified and submitted to the registry, like that's a good checkpoint for, hey, let's put them up. So we did some thinking about how to fit this process into a real sort of workflow, at least, very much tied to the academic research environment. And then also thinking about implementation, we tried to make a derived data registry. It's very simple. It essentially is that links to a GitHub repo that has this folder structure. It just has a log, some code, and the data. I think the biggest thing to think about is the log. And so we tried to actually fill in the fields that we suggested people fill in for a variable that's under development, and then one that's actually been registered. And this is all in the JSON format because we were aiming for that compatibility with PsycDS. But this could just as easily be a spreadsheet or CSV file or anything like that if you're working with a group that's not into JSONs. The other components are the code and the data. And that's about it. I'll turn it over to Megan to just wrap up really quickly.
Yeah. So we think we're helping people solve a very real and tangible challenge that often wastes time and money and effort. But we're also trying to make this a gateway into some more open science and reproducibility practices at the same time. So here are a couple of baby steps you can take to change your processes to help you in the long run, but also to maybe eventually start submitting to journals where you're required to share your code and it's ready for, lots of eyeballs all of a sudden. So we we have to acknowledge we have not actually implemented this in practice with real data that we work on for our research. But we would love feedback if people want to try this or doing something similar. So any questions, suggestions, feedback, please create issues on our GitHub. It's No Science is an Island. And thanks again for your attention and for everyone's attention tonight. I know there's a lot of presentations, but I just want to end with a huge thank you to all the Neurohack organizers and instructors who've made this a great experience, and we've all learned a lot from. So thanks.
Thank you.
Right, next is the bids in spec. Bids in spec.
Yeah, we're here. Sorry.
Sorry, I was just trying to figure out how
Sorry,
to share my screen.
Okay, great.
Okay, so I think we're all set.
Let's see that I can get you on the screen though. Okay. All right.
All right. Can you hear me?
Yes, we can hear you
Okay,
well.
great. All right, so hello everyone. My name is Nancy, and along with Ashley, we are the team behind the creation of Bids Inspect. Next slide. Oh, thank you. And so Bids Inspect is a Python library that will help researchers summarize, visualize, and validate their bids datasets. So some of the capabilities, the main capabilities for Bids Inspect is that it will generate a summary report for the bids compliant directories. And we believe that this addresses an important need that has arisen from the increasing popularity of automated workflows. So we hope that the summary reports created by the Bids Inspect will hopefully encourage researchers to examine their bids datasets prior to jumping into pre-processing steps by providing them with a report of features like scanner type, magnetic field strength, acquisition time, and resolution. Additionally, another capability of Bids Inspect is to validate a bids dataset against an acquisition protocol. And we think that this will help address a problem of heterogeneity across MRI session acquisitions within and across institutions. And some problems that arise with this include incomplete scans and variations in scanning parameters. And so with Bids Inspect, we hope that researchers can basically validate their data set against an acquisition protocol using features found within JSON sidecar files, as well as nifty image properties such as image shape and resolution. And in order to implement this tool, we decided to go ahead and make it a standalone tool instead of a bids app or a plugin to an existing bids conversion tool. And the reason for that is due to the interest of time and the complexity of what that would look like.
So I'm going to go ahead and talk a little bit about some of the implementation details for the report generation portion. I just wanted to quickly give you guys a little example of what I'm referring to when I say generating a report. So this is an example report from fMRI Prep. And so this report contains like summary details about the fMRI Prep directory, and then it also contains visualizations of scans within the directory as well. So we were inspired to take the same idea, but apply it to a raw bids directory. And so what we did is we set out to create these reports. And so we found this Python package called NiReports, and which unfortunately has very little documentation, which made this whole project very tricky. But NiReports basically consists of two components. You have reportlets and an assembler. And so the reportlets you can think of as like individual, like a summary table or an individual figure that would then go into the final report. So the reportlets are basically all of your like subcomponents of the report. These tend to be .svg files or a .html file. And then you have the assembler, which basically combines all of those reportlets into a single final report. And it does so with the help of a report specification file, which is basically just a config file that is a YAML. And this report specification file contains all of the exact details and instructions on how the assembler should combine the reportlets to produce that final report. So we're like, okay, we've kind of got the gist of how the NI reports works, but then we realized that creating these reportlets is non-trivial. And so we had to figure out a good way to actually generate those figures. And so then we realized that we actually had to create a NIPipe workflow. And so that's what I spent the majority of my time doing during this hack week, was just creating that NIPipe workflow, which creates the figures, which will then be used in the final report. So here is a graph of what our NIPipe workflow looks like at the moment. It is small and definitely still a work in progress. Just to give you some background information about NIPipe, it is a Python package that is used to create reproducible neuroimaging workflows. As you can see here from this workflow graph, it is a directed acyclic graph. So you can see it consists of these nodes, and you can see that the arrows here indicate the flow of inputs and outputs throughout the graph through those nodes. And each node will have an interface, and that interface can wrap many different neuroimaging tools, or you can create your own. And so this is really, really powerful, because this node could have an interface that wraps FSL, that wraps AFNI, that wraps FreeSurfer. And so it's really a unified interface in order to combine all these tools. So just to kind of give you a little background about what NIPipe is, I highly recommend people look into it as well. But yeah, so back to our specific NIPipe workflow. It consists of only 4 nodes. So you have an input node, and then the second node is going to be a bids grabber. So you give it a bids data set, and then it goes in and extracts different scan types from that bids directory. And then those bids scans then get passed into 2 nodes that do some figure plotting. So this is the same workflow. It's just represented in a more detailed graph where you can see the exact files that get passed. So you can see that the bids grabber takes all of the T1 images and passes it to the node that will plot the anatomical scans. And then you can see all the bold files from the bids directory then get passed to the fMRI summary node, which will also produce a figure based off of that. So this is what the outputs look like. They are not the prettiest figures at this point. It is still very much a work in progress, but this is the mosaic.svg file that gets produced by this plot and nat node. And then this is an fMRI plot.svg file that is produced by this fMRI summary node. So this is going to continue to expand as we add more interesting figures and make these figures also more pretty and useful. And so this is definitely like a work in progress that'll continue to evolve. So we decided to wrap our NIPipe workflow as a command line interface. So this kind of just gives you an overview of it at the moment. So it takes in a bids directory, and then it'll run that workflow and produce those figures in an output directory for you. This will continue to expand as we add functionality, or we could even just turn this into a bids app as well. So yeah, basically, this week we spent implementing this portion of this figure. So this is the NIPipe workflow that we've now created that has generated some reportlets. And so those 2 nodes that I showed you, the plot and nat and the fMRI summary, are both NIPipe interfaces that were created by NI reports. And during this week, I've also started to create a custom interface for NIPipe that will plot the BVEX for the diffusion sequence on the sphere. So like this is an example of what that could look like. And so that's like still a work in progress. So hopefully that'll be able to get wrapped up soon. And then the next step is obviously to use the assembler to actually combine the figures that we've generated with our workflow to create that final report. So we hope to continue working on that in the future as well.
So prior to Neurohack, I was unaware of bids and the different many bids apps and libraries. So I really had to get up to speed and in order to be able to contribute to this project. And so I acquired that knowledge throughout the week. This included general knowledge on bids, so like looking at the lectures from last week and then all of the different resources that bids has available. I also learned more about different types of bid converters in order to think more about along with Ashley about the implementation strategy for BIDS Inspect. So I went through documentation, tried to like get my head around how that would work for our tool, and also learning more about tools like PyBids and eBabel. So in order to test our pipeline, I helped with the data selection using OpenNeuro and the BIDS examples repository. The OpenNeuro data sets that we selected are going to be used for the actual testing of BIDS Inspect. And the BIDS example repository data sets are useful for soft testing because they don't have the big raw data files. And so in order to get experience with BIDS, I validated the data set using the BIDS validator via the command line. And I also queried the BIDS data sets using PyBids. Throughout the week, I got a lot of experience and learned about different things, and through this we were able to kind of graph out the next steps that we need to take in order to implement the next capability, which is the protocol validation. And for that, we need to design a feature that helps us get image properties with tools such as eBabel out of like Nifty files, or like extract things like image shape and resolution, as well as extract features of interest from Nifty files like that will be a part of the protocol validation config files. Oh, next slide. So prior to ending our talk, I'd just like to direct you to the wiki, where we documented all of the process towards designing BIDS Inspect and the different types of resources that we looked at, and a link to our GitHub. And we'd like to thank you for your attention and thank Dr. Noah and Dr. Ariel for allowing us to be a part of Neurohack Academy. Thank you.
Okay.
Thanks, that was great. Next up, Think, Share, Care.
I was, but I guess it just crashed. Oh, okay, yeah, that makes sense. Were you on the Zoom? Oh. Yes, I have mine.
You don't actually need to join out at all. All right, cool. Yep, sure.
Yeah, so our project is an interactive web app called Think, Share, Care. Okay, yeah, so Think, Care, Share, and as it says here, it is an interactive web app for mental health clinicians with the purpose of integrating quantitative data into clinical decision making. All right, I'm gonna just control this. Okay, so kind of to break down what our app actually does, the first component is Think. So our app allows community contributors with patient data, including mental health care clinicians, hospitals, and researchers, to create models that predict outcomes of interest, which could be anything from potential like current diagnosis, diagnostic subtypes, or treatment responses with, of course, the necessary data put into the server. The next step is, or the next component is share. So after these models are created by contributors can then submit the predictor weights for their models without actually sharing any PHI, which is pretty essential to kind of sharing this information. And yeah, you don't usually want to just share all your PHI, but kind of getting together or integrating different models to improve some higher level model with all the data would be helpful to theoretically improving the accuracy of these models. And to do this, we used a meta-regression technique, which we'll explain in a second. And the third component is care. And this is kind of the clinician-facing side of the app, where clinicians can input their patient information into the tool and generate a score that will kind of help them assess their patient's odds of a specific outcome. Again, this would be completely done on their own computer and would not be submitted to a server or anything like that.
I mean, is that a crazy thing to?
Wouldn't go that far down.
All right, yeah, so that's basically kind of the background of what our app does. An important note kind of before.
I put it so that the sound will pass through still, so I can hear you. Yeah.
All right, cool. So that's what our app does. An important note before going any farther, an idea that we really want to highlight while going through this presentation, which is really well consolidated in this quote that we actually saw in the population health building here at UW, is the world cannot be understood without numbers, but the world cannot be understood with numbers alone. And while we think that this tool could be powerful with enough data to help clinicians and inform or use quantitative data at least to help inform decision making, there's also a lot of other information that goes into making clinical decisions. So we just wanted to highlight that before proceeding. So I think to help illustrate the utility of this app, one thing that's important context to understand is like background of like some ways that clinical decision making is currently done maybe in the clinic. So to do that, I'm going to just talk briefly about the DSM. So the DSM is the Diagnostic and Statistical Manual for Mental Disorders that most clinicians use to make diagnoses and kind of steer treatment. So here's just a little blurb from the DSM under major depressive disorder, specifically the risk and prognostic factors. So here I'm just wanting to illustrate that we do know risk factors for disorders like major depressive disorder. For example, that says like neuroticism is a well-established risk factor. So personality characteristics. There's also environmental factors that we know can play a role, like adverse childhood experiences, particularly when there's like diverse kinds of experiences. There's also genetic and physiological differences, as well as comorbidities, both mental health and physical health comorbidities. So this information is given to clinicians and we do take that into account, but the purpose of this app is kind of trying to make it more quantitative. Or yeah, because for a couple of reasons. One, like obviously considering all this information can be a lot on cognitive load side. Like we can't possibly, if our client or patient has like multiple of these risk factors, how are we really, computing in our head how much more at risk they are? That can be like rough on the clinician and difficult to do. And there's also the possibility of just like clinician bias, like clinicians will have different experiences and that will inevitably kind of start to drive how they're perceiving new patients and developing and planning treatments. Just some brief, like obviously these kind of prediction calculators, like people have tried to do this and not that our app doesn't have limitations, but these are just some limitations that maybe other calculators have that our app addresses. So the first is data availability. The idea of the app is that it would be a way for clinicians, researchers, and hospitals to allow a lot of data to be integrated into the models. So ideally, it would make a lot of data available. Also, a note on like feasibility, the NIH. A lot of studies funded by the NIMH already require you to submit your data kind of to their archive. So if this could be paired like formatting wise with something like that, I think that could allow for a lot of data to be available and a lot of data to kind of go into these models. The other thing is ethical and privacy concerns. So as I had already briefly mentioned, no actual data goes up to the server. It's only these like model weights, which we'll talk about in a second. So that helps to eliminate some ethical and privacy concerns. Also model interpretability. Some of these models that have been used are fairly complex, and like clinicians looking at the models maybe might have a hard time like understanding exactly what the models are doing, and thus potentially could have less trust in, you know, what the model is actually giving to them. Our app uses just linear and logistic regression, which like has its own potential limitations, but at the same time could be potentially easily explained to a more lay audience and may encourage more trust in the models. Yeah. And the last thing is updating. So ideally, this app would allow for updating of the models so they could have endlessly like more data to kind of inform the model predictions. Okay, yeah, so with the background of the app, now Dylan's going to tell you what the app does specifically and how it actually works.
Thanks. Yeah, so I can kind of speed through these, but we just, you know, we have our X and Y, and we like to generate these prediction models, and these data are sort of used to inform that DSM risk and prognostic factors piece that we showed before. But it's hard to tell, like, if our patient is here on the x-axis, where according to this prediction algorithm or just linear regression, where do they fall, where are they expected to fall on the y-axis. So the basics of the app is you have two contributors, let's say. Contributor one puts in a model with an outcome, and they have variables A, B, C, and D. Contributor two has the same outcome in their model with these 5 variables, 4 of which are overlapping. So when they both contribute their models, the overlapping variables get fed, specifically the overlapping weights get fed into a meta-analysis, with which the weights are updated. And then in addition to that meta-analysis, we're also storing that contributor two's model. And then that gets stored in the server, and then contributor 3 comes along and has the same outcome, and all these variables, some of which are overlapping. So the first meta-analysis gets updated, the second one gets created, and then again, we're storing that individual model with more variables. Okay. And now, theoretically, it should work on the internet, but I don't trust the internet. Okay.
Shared tab.
Oh, shucks. Okay.
You're on sharing now.
This is our app. So there's these 2 tabs. The contributor who inputs the data and generates these models, and then the user who's the clinician. To go in, you can choose your test or your data, and then you select the outcome, and then you can select the predictors that you are in, that you have and want to contribute to this model. So then you will run the model, and then it gets saved to an AWS server, and then oh boy, let's see. Then you can rerun the app. Ideally, you wouldn't have to rerun the app. You would just be able to click through. But yeah, we only had 3 days. So then the clinician goes in and says, "Hey, I have this client, and I have some really basic demographic variables, and I'm curious. I have a differential diagnosis to make between ADHD and like ODD or something." So you select the outcome, you search the models. There's no meta regression because there's only one model that's been input already. So it's just saying that your model is just based on one data set. So then you can input their age, random numbers, and then
you get some scary errors, which maybe will be fixed one day, but you also get a predicted value. Very cool. This is log odds, so you still have to do that math to convert it to an odds ratio. But then, anyways, then you can do this again, and the next time you do it, I promise, instead of using the weights from the individual model, it pulls from the meta regression. So then your weights are updated and more robust theoretically. Yeah. I would show you that, but we've been talking for a bit, so I won't show you that. But I promise it works. Okay. And now, real quick, we'll just talk about the limitations. So there are obviously a lot of limitations here. One of which is we're determining an overlapping model by which variables are shared between contributors, but realistically, if we're like, do we want to run a model that was tested on adolescents and then update it based on a data set of older adults? Probably not. So that's one thing to consider. We don't really test for model assumptions, which is obviously a big no-no. Future directions. Right now, the only measure of trustability that we have in these meta regressions is I-squared, where higher I-squared indicates more heterogeneity of the effect size. There are potentially better metrics of this. Also, we're not considering interactions between variables. These are just like very basic models that we're creating. Model selection, which I'm blanking on what that means.
But we could incorporate something where the models with the highest R squared are the ones that remain in the server and the other ones are thrown out, but we didn't do that. And then the potential for overlapping subjects.
Yeah, so if two contributors submit a model and the models have overlapping subjects, there's no way of knowing that because we're not storing the
IDs, like
Yes.
the identifiable information.
And also, so there's a similar thing that does this called CoinStack by Enigma. What they have you do is create a model, and then when you create a model, a consortium gets built, and then users can sort of buy into that consortium and contribute their data. And that's good because it makes sure that the models that are being input are robust and not bad. So our model doesn't really have any barriers to entry, which is good, but also bad because if someone puts in bad data or bad models, then there's no way to sort of account for that. So yeah, future directions. And yeah, really enjoyed this time. Thanks. It's nice to meet everyone and have you have us here. So I appreciate that.
Thank you.
Thanks.
All right, on to the federated learning team. Representing the laptop. Put it down here and then join Zoom.
And on Zoom.
Follow your color and close.
Oh, no. I probably did. You are so right that I did. Yeah. I should have never included it. Oh, gosh. I also got kicked out of Wi-Fi. Right.
Don't join audio.
No. Where do you do the presentations?
All right.
Hi, everyone. We will be presenting on federated learning with healthy brain network data, integrating free surfer metrics and age-based stratification using the Flower framework. Thank you. So as far as our outline goes, our aim, well, big central aim was to employ Flower, a federated learning framework that allows for the distributed training of neural network models, as well as scikit-learn, to predict brain age using brain metrics from the Healthy Brain Network. So scikit-learn, it's a Python package that itself doesn't have federated learning programmed into it, but it is really, really effective in combination with Flower at creating this federated learning model. So problems with centralized machine learning approaches, which is the reason why we decided to go with federated learning, is it usually requires a central data repository, which takes a lot of infrastructure and a lot of money. It also does not scale well to large data sets typically. And there's also a single point of failure risk, meaning that one problem is going to lead the whole system to shut down. And that's the case when there are data breach and privacy concerns involved. So why federated learning? Simply because you can have multiple data contributors. There's no real raw data exchange, which limits those privacy concerns as well as data breach concerns, and it's decentralized. Learning how to use technology. The Flower framework. So federated learning requires an infrastructure to transfer machine learning models, train and evaluate them on each local data set, and then aggregate those models. Flower is an open source platform, and it provides the infrastructure in a way that is unified, easy, and scalable. So as you can see from the diagram here, there are multiple of these servers. Each has their own data. And the great thing about federated learning is that all you have to do is get the computation, and you can run it yourself on your local drive. So as you can see here, the results then go into the server, and that's how it continues to aggregate over time. So federated learning with Flower. The first step is initializing the global model. And after that, the second step is that you send the computation or the model itself to each of the clients. The third step is that the models are trained locally on each group's drive. And the fourth step is the model parameters are sent to the server and then officially aggregated, the results.
So the first thing we did was to look at the documentation for the Flower framework, which is a Python package basically. And they have a tutorial where they use the MNIST data set, which is a very standard benchmarking machine learning data set. And then they use a logistic regression model from scikit-learn. And then, so we tried to run it. Yeah, it's got some problems because their tutorial code was not entirely accurate. Fix the problems, managed to run on everyone's laptop except mine because mine is too slow. It wouldn't run the import statement, so that was kind of sad. Yeah, and then so basically there's two scripts, one to run a server, and then for this tutorial and all of the things that we did after that, we basically took one data set and then partitioned it into a different data set to simulate a distributed learning environment. So it was still the same data set. There's other issues related if it's actually data from different sites or different clients is actually coming from different sources, and there's other processing things that need to be done to make sure that the data is consistent. So we didn't really deal with that. So we run a server and then we run a bunch of clients and they connect to the server, and then the federated learning thing happens. So then after that was I think the second day. After that, we adapted the tutorial to run our own experiment, which is using healthy brain network data, where we extracted the free surfer features for cortical thickness and subcortical volumes, and then we tried to use that to predict age. The age range for the data set was 5 to 21, and the sample size that we had was 223. So not huge, but like we thought it might do okay, maybe. So our goal was to kind of explore federated learning and different scenarios that we could think of, specifically by splitting the data set in different ways. So first we tried equal sample sizes, and we thought the easiest case would be if the distribution of age, sex, and site was the same across all of the partitions. And then we tried what if all the different sites have different age ranges and what would happen then. And then finally, we tried what happens if the different sample, we have different numbers of partitions, and they also have the different sizes. So now we're gonna run a demo for
how it works.
Yeah.
So I'm gonna stop sharing, and then I'm gonna share the VS screen. Oh my God, I'm scared, but it's okay. I promise it ran like 2 hours. Okay, I'm in Zoom. share screen. Cool. So, basically, the way we demonstrated multiple clients is just starting them from our own computer. So this is all based on the Flowers framework. First, we start, oh no, we don't want to run everything. We're first starting the server script with it's in the path.
Dash data. And then you remember the name of the traveling?
No, I do.
Yeah, that's it.
Right? And then the server is running, it's waiting for clients, waiting for 3. So we have to run the clients 3 times.
Actually, this is me waiting for one.
Why is it just
Sorry, because we didn't set, I set them, yeah.
Yeah.
Okay, sorry.
Okay. What's the
Dash
argument?
min.
Okay. So
Sorry.
now it's waiting, actually waiting for 3
Yes.
clients. I'm gonna run it like this. This is, yeah. Okay, so now this is the first client. Supposed to send. Oh, also, we set it to do 5 rounds of the sort of aggregation altogether. And then this is waiting for the second client. Or sending the second client.
That didn't work.
Didn't work. And is it because of? Well, it's not giving the data.
This is also a real life demo to our brainstorming process.
Is it also data?
Yeah.
That worked.
Yes. I think so.
Cool. I think so too. And then.
Yeah.
And
so it's quite fast. And it basically everything. Once it waits, the server waits to receive everything, and then it just
runs.
Yeah, it just runs.
And we are printing like our accuracy and loss.
Well, accuracy
measures.
is actually the R squared.
It's
We
just
didn't
R
change
squared.
The labels from the tutorial.
But we have figures to show
Yeah,
what actual
yeah.
results we got from
And-
the different runs.
Yeah.
But
don't have much time left, so
we don't have that much time left.
Yeah, we're at 8 minutes.
Okay. All right, so.
Yeah, so real quick, this is just how we split our data for a normal distribution. This is just the data we use stratified. And then we also did grouping by age. So we had 3 groups. The age ranges were from 5 to 21, and we grouped them up so there was a different distribution.
So here are our results. This was on 80% of the full data, and there were no partitions, which means that 20% was used as the test data. And our mean square error was 5.06, and our R squared was 0.6, so it did pretty well. And you can see the weights or coefficients on the plot of the different free surfer regions. And the blue bars are the positive coefficients and the orange are the negative. And it turns out the brainstem contributed the most. And then next slide. Just to visually show the brainstem in case anyone forgot where it was. It's there. Because the rest of the regions were aggregates and basically the whole brain, so we didn't plot those.
Oh, okay. All right, also just a little note on our models selection, like model process. Just want to touch on it. We use lasso regression. I don't know if everyone's how much people are familiar with it. It's basically a method where when you have a lot of features that you can use to predict a outcome, you first train the model to like choose which features, or not first, but like there's a like the equation includes a certain loss sort of regularization to choose which of the features are important and like drops the features that aren't important. And this is the one that worked, that we wanted to go with because our data set was only like 200 people. And we had like 100 something features. And we also know like a lot of like age and brain can be predicted. So that's why we wanted to choose this relationship. And that's something that was like novel because we wanted to see the results of federated learning. Okay, so I'm very excited about this part. So we didn't get to do all of the tests we wanted to do. There's a lot of ways you can play with this like federated learning framework, but we got to do some. The first thing we wanted to do was look at how different number of splits affect the outcome. So you already saw that for our training on the whole data set, the R-squared was like 0.64. Every time you split, think of the whole data set becoming that much smaller for each little split, right? So I'm sorry about the y-axis. It is not very organized because I didn't have a lot of time to organize my variables there. It's very, very last minute thought, but if you are able to look at the last 3 splits and the top 4 splits, the 9 splits, you can sort of see that there's like not that much of a difference between how many times you're splitting the data set, which I think is very interesting because when you get to 9 splits, each one has like 20 something people. So I think that's pretty interesting. And then the other 9 splits, the small, that part is not something we've interpreted yet, but the results aren't very different. Just for clarification, that's where we split the data set into 3, but one of the 3 was much smaller than the other 2. So it was like maybe a 1/9 and then a split of 2 with what was left. That's where I wanted to test the noise stuff, but we didn't get there yet. I wanted to like poison the small one and see like how well it would hold up to the poisoning. It was maybe later, maybe this evening, I don't know. The interesting part is if you look at the one in the middle, the 3 splits grouped is where we had the 3 by age group. And you can see that that one performs really, really badly. So perhaps this tells us that it doesn't matter as much how small the data sets are, but if the data sets individually cover a range in which the relationship that you're looking for is quite different, which we know changes a lot because brain growth is not linear with age. The relationship between 5 years and 21 years and like the growth of the brainstem, it's very different relationship if you look. And we're running like the basically linearized regression. We're like accounting for that. So if we're thinking about like collaboration aspects and all of that, I think it's very interesting to think about if you have multiple sites, but their sampling is different enough from each other, and you don't know what variables are going to matter. That's something to think about. But you don't necessarily have to share data. You can still have small data sets, but it's important to think if these data sets are similar in their characteristics that matter. And then this is just the inverse of that. So I'm gonna skip this one. All right, do you wanna?
So here we've basically combined kind of 2 things that Erin did, but we're doing it across. We've the loss in accuracy. We're going to show you one where we had a small number of groups. So this is 3 groups. Just a little note that you might see there on this side. For what we're doing here, good models have higher accuracy and lower loss. These are not to scale. So as you can see here, we're doing fairly okay across each round. And again, here on the loss, pretty similar across each round. Here we see now we have 6 groups. So this is our intermediate size of grouping. What we see here again is that across the rounds, we're getting very similar accuracy, very similar loss. And as you remember from Erin's little graph, these are very similar to the 3 groups that we had initially. And finally here we have the biggest split that we did. So this is the 9 groups, all equal sized, all very similar distribution. And as we see once again, we have very little variation there on each of the rounds. We're getting a little bit noisier in the loss area, but we're still doing pretty good. And this one here is where Erin's last one came up. As we see here, we have grouped by different ages. So we have 5 to 9, age group. We also have a 9 to 12.5 age group. And then we have quite large age group at the end because the density is lower across those years. So basically there is 57 individuals in each of these groups. But as we see here, because of the fact that there is a low density in the third group, we actually see that it's that little one here. That was our client that had the 12.5 to 21 years. So as we can see, once they're kind of similar distributions, we're doing fine. But we can see here for the accuracy for the group that has a lower density and a broader range that we're not doing quite as well.
Okay. Hi, everyone. I'm so sorry. I think we've kind of ran over time a little bit. So we've had lots of challenges, lots of learning, and it's been an absolute fantastic experience, and we've collaborated so well. So I'm just going to like briefly skip this kind of stuff, and we will put up our slides on GitHub. So if you want to finish what we've done, these are just some group photos that we took. We had a lot of fun, and it was an amazing experience. And oh yeah, sorry, forgot your review. And these will also be up our GitHub link and everything on those slides. Thank you so much, everyone, and for listening to us.
All right. We have two more groups. First, the Neurohack group. Yep.
Ready? Awesome. Hi, everyone. So yes, we are the NeuroNav group. We are the group that was creating or using the Voila and Voici Python libraries to create an interactive web app that explores the HCP young adult data set. And we're really excited to share with you all what we've ended up with and how we got there, which proved to be perhaps a little more challenging than maybe we were initially led to believe from the Voila tagline, but we're really excited with what we ended up with. So yeah, we'll show you all what we have. Our initial goals for this project, we had two kind of main learning goals. I think first was to really get experience with these two packages, which none of us had ever used before. Basically, these two packages enable you to turn a regular Jupyter notebook into a like standalone web application that you can just run in like a browser. And the second kind of goal we wanted to learn about was the Human Connectome Project Young Adult Dataset, just being able to explore this data set. And we thought it'd be a really good one to use for sort of this activity because it is such like a large set of data with many different kinds of data in it. So we could plot theoretically many different things in our web app.
So as we were planning this app, this was sort of our vision of how it would work. So as Sophia mentioned, the Voila and Voici packages are built upon a Jupyter notebook. So we thought that we would have our notebook where we build the features of our web app using Python. And through the magic of Voici, this gets transformed into a web page that is deployed and hosted using GitHub Pages. So another aspect was how do we get the data from the Human Connectome project to be accessible for our web app? And so we were successful in being able to pull data from an open bucket on the AWS S3 server into our notebook to display. And we were also able to build a file upload widget so that if the user has downloaded their own version of the HCP behavioral data set, they could upload this to the app and have it displayed. So it was very exciting to get these features working as we were testing them in our notebook locally. But then when we tried to go to the webpage version, it turned out that neither of these avenues for accessing the data ended up working. And we spent a lot of time trying to figure out first of all, that this didn't work. But then second of all, to understand why. And where we kind of landed is that when you're using a web browser, there are often security concerns when it comes to trying to pull data from different locations, and we just weren't going to be able to overcome those in order to access the data on this website. So our solution and the demo that we're going to show you today is based just on an interactive Jupyter notebook alone. But ideally, you know, as a future direction or goal, we would love to have this actually converted to the web page.
So yeah, just to show kind of how far we got in terms of the actual web app side of things. So this is a screenshot of our GitHub repository. The link that would actually point you to the web app is both in the kind of README section down below. It's also on the side panel under About. So if you were to click on that link, it would take you to sort of the landing page. It shows the notebook that's able to be run kind of interactively. So you double click on the notebook, it would start up the kernel, and then it would pull up the actual web app, which looks something like this. It starts off showing you an interactive plot on the bottom. That's for the neuroimaging tractometry plots that we were able to make. That's just using some data derivatives that we temporarily put up on the repository since we weren't able to access them through the S3 bucket. But if you were to have your own data, like Rohini was mentioning, there is a little upload widget at the very top that would populate the top two panels, which are initially empty. But unfortunately, when you click on that upload widget, the file doesn't populate anything. So we'll now switch to the actual Jupyter notebook to show you all we managed to build.
Yes, so I let that run beforehand. It doesn't take long, but essentially I could let it run anew, but just so that we're sure that everything is actually still working. I guess the benefit of using it in YC would have been that you wouldn't have even seen this whole Jupyter site and you wouldn't have seen all this code. So it would have been more beginner friendly. But for someone who's ever used Jupyter before, it probably shouldn't actually be that much of a difference because you just have to scroll a bit. And then you land here and you should hopefully recognize this because it looks a lot like the screenshot we just showed you. And it's also still empty and it also already has these plots. And there's an upload button. And that should, hopefully, this is the behaviorals data file from the HCP website. You can download that yourselves. It's just not on any buckets. And here it works. So on the left, we have some demographics info. And basically, it just shows you the age brackets. So the data is represented in age brackets for data privacy. They don't give the exact ages of participants. And yeah, you can basically just see how many people are in each of these age brackets. You see there's a 36+ group, but it's really very, very small. You can also see the gender distribution here, female and male. And you can select ages. It'll show you what you selected. It will not yet show you the gender distribution for that bracket. That is work in progress. That's the demographics info you can get.
So the next panel shows our behavioral data. We have two dropdowns here where you can plot different variables. We also decided to add these toggles that also let you subset the data based on either age groups or sex. There's also an option to show the regression line. Yeah, these toggles currently don't actually do anything because we didn't have enough time to incorporate them into the rest of the data, but yeah.
Then for the final panel on the bottom of our webpage, this is where you can explore some tractometry data. So right now, this is just hard-coded for a single subject, but another future implementation would be to add a dropdown where you can navigate through different subjects' data. But on the left here, this dropdown shows you different tracks or different pathways that you might want to plot, and you can toggle between them. This one nicely animates, which is another feature that we had working at one point up here, but is no longer, but that's all right. And for each of these tracks, there are 4 different actual diffusion measures that you might want to plot. And so you can go between them here as well. So I think that wraps up our demo, and then we have a couple more slides. So for future directions, one thing we considered was reporting our issue with the file uploader bug to Voici to see if anybody could figure out what was going on with that. An alternative was to also implement this with Finder or Docker, which could possibly help us bypass this file uploader bug. Also adding more interactive elements between the behavioral data and demographics, getting those toggles to actually work. And adding the ability to toggle between subjects for the track plots as well, since we only had one subject currently. And then also adding a key for the variable names, just so people know what they're looking at with the data.
Yeah, and then some takeaways from this project, aside from it having been a bunch of fun. So thank you all for that. And yeah, there's just lots of roadblocks to accessing data through web apps, whether that is from a bucket in a cloud on AWS or from your own local device, things that generally work in Jupyter and probably might work if you host Jupyter on a web server or all that, but like hosting it in the browser locally to run on the client side so that the data isn't ever actually uploaded anywhere presents a bunch of issues. That was an interesting learning with a lot of debugging. And also important to specify package versions in the environment.yaml, which for now was not an issue because we're coding this within a week, so nothing's really changing, but we tried to access a couple of other projects that had previously successfully implemented upload buttons in Voici, and except for one of them, they all would not even start because presumably they were 2 or 3 years old and their environments were not up to date and they would not compile anymore. So keep that in mind if you want to future-proof your projects. And I think one of our main learnings really, aside from the specifics of this project, has just been to work with GitHub. We had lots of fun and trouble branching and merging and sometimes rebasing and all the other things. Yeah, so I guess just thank you to all of you for all your comments during lunch, dinner, and anywhere in between, and the instructors, of course, for all your helpful guidance throughout this. Thank you.
Thanks a lot. And now for the common communication. The ED-based classification of behavioral states. Anything. Are you on Zoom? Yes. Are you sharing your screen?
Not yet.
Okay. Whoa.
Sorry.
You
just drag it, right?
You can do that where you can share the whole slide.
You can share it all.
Share the whole
Like if you
share the whole thing.
stop sharing, share.
Oh, just got screen.
Screens.
Share all thing.
That should work. That's fine. Oh, screen sharing is possible.
Because
There we go. Oh, all right.
How do we do it?
Don't have to.
Good.
All right, everyone, welcome to our presentation. We think have been the only group working with EEG data, something that was also pretty new to us. We've all had a abiding interest. And to get us started, we found a publicly available data set and dove right in. So first, we're just going to cover a little bit about the data set and the tasks that the volunteers have reported during, and we'll go into some of our analysis techniques and finally see how far we got with our analysis. So first off, we found this really rich data set on a website called PhysioNet. And in case anyone is interested, they actually have several different physiological data sets, not just EEG, but EKG and other cardiac signals as well, other neural signals as well. So lots of data, software. They also have a challenge every year. The incredible thing about this data set is that it was pretty huge. So 109 volunteers recorded both 1 and 2 minute recordings with this 64 channel EEG set. And the main, very simple task that we were looking at was, could you distinguish between the eyes being open and eyes being closed? And so for part of our pre-processing, we did something which is very standard in the field, which is extracting the different band power for alpha, beta, et cetera, and also some averaging and other pre-processing techniques.
So one of the analysis methods that we did was random forest. So this is a learning method that builds multiple decision trees during training, and it merges them to get a more accurate and stable prediction. Each tree in the forest is trained on a random subset of the data and the features, and the final output is based on classification or regression. So they're just taking the average of the different values of, or the average of the different like sub predictions that they do from the data set. So some of the pros of this is that it is pretty robust. It indicates the importance of features, and it's a versatile model, and it also handles missing data via proximity. However, it can be computationally expensive, memory intensive, and there is a bias to the imbalanced data. So we had 100 estimators and 218 samples total, and our average accuracy on this was 73.89%, so not horrible. And if you look at the confusion matrix, it kind of like, they go diagonal. So the true value for eyes open was 80, and the model predicted 81. And the true value for eyes closed was 28, and the model predicted 29.
Yeah, so gradient boosting machine is a technique that's very similar to random forest, but it builds the trees sequentially, while like each successive tree kind of builds on the errors of the previous one, and focuses on minimizing loss function, usually via gradient descent or some other kind. Pros here are high accuracy, especially because it builds on the previous iterations. It kind of indicates the importance of features, and it's a bit more customizable, but also like random forest can be computationally expensive. And because it's incorporating past iterations, is more prone to overfitting. Here we got around the same accuracy with 100 estimators, around the same precision. Really not much improvement at all.
And then we also did recurrent neural networks. So this is a type of neural network that's designed to process sequential data. They have connections that loop back on themselves, and it allows them to maintain like a memory of previous inputs. This is great for sequential data, and there's temporal dependency. It also has a flexible output. It can handle variable length sequences. However, it is a bit more complex and harder to interpret, and there's limited long-term memory with these models. So for this, our accuracy was again around the same. It was 73.89% using the RNN. And again, you can see the confusion matrix, which is not as good as the previous ones.
As for the layer architecture, RNNs traditionally are quite bad with long-term memory. Usually you're fitting something like time series data to these. In this case, we're not fitting the raw EEG data, we're fitting the scalar values extracted for each channel. Here we use 264 LSTM layers. The long short-term memory LSTM was an addition to RNNs that allowed them to handle, to have a longer short-term memory, as the name says. We have a 64 dense layer, which is, so you have your input layers, the LSTM layers, and then the dense layers are fully connected to every previous neuron. We use some dropout layers, which on each iteration randomly choose neurons to just disable, and this helps to prevent overfitting. And then we use another layer of dense neurons as our output activation, as our output and prediction layer for with an activation function. And finally, we fit convolutional neural networks, which are more traditionally used for image classification. We didn't really think this would work very well, but it seemed to. They're a deep learning model that's used to process grid-like data, and they use convolutional layers to sort of learn about the spatial hierarchies in data and exploit that for prediction. As I said, they're great for image classification, object detection. They do automated feature extraction with the spatial hierarchies, and they can handle transfer learning as well. The cons is that they require large amounts of data and are often prone to overfitting, as well as sensitive to input variations. And here we used a conv2D layer, a max pooling layer, which downsamples the data, and then this is flattened into a 1D vector in the third layer. Then we fit the dense layer as well as in the previous RNN case, and again use a dropout layer to sort of bias against any overfitting. And finally, an output layer as well. And here we were able to get around 80% accuracy, which was quite cool. So here's sort of a summary of all prediction accuracies, pretty much all around the same, around 73% for the random forest gradient boosting machine and the RNN. And then CNN kind of outperformed. And so we kind of sat with that and asked, why does this seem to perform better? The conv2D and max pooling 2D layers are unique to convolutional neural networks. The first capturing spatial hierarchies, then band power features, and then this isn't explicitly modeled by something like an RNN. And then the max pooling 2D layer reduces the dimensionality of the data and maybe prevents overfitting. We thought that the CNN at least may outperform traditional ML models like the random forest or GPM because these latter models require averaging out the differences in the band power across channels. With both the RNN and the CNN, we were able to feed 64 channels with each of the 5 bands. And lastly, we thought, if we used the raw EEG data, maybe the RNN would have performed better than the CNN. You want to say some final words? Here's some references.
These are references.
We just wanted to thank everyone who helped us. As we know, there were a few twists and turns in our journey to this project. Special thanks to Ariel, Noah, Ian, and Kelly for all the help setting up the Jupyter repository and getting us over the final finish line.
Thank you so much for all these great presentations. We will now take our farewell from our friends on Zoom and to see you sometime soon. And those of us who are here, we can slip over there for dinner, which is waiting for us.